Welcome, discoverers. Today we are exploring a hypothesis that could reshape our fundamental understanding of insulin resistance. For over 100 years, we have believed that insulin resistance was primarily a cellular issue. But what if a key piece of the puzzle has been missing all along? What if insulin is being lost before it even reaches its target?
This is great because I'm learning straight away. Don't know what insulin resistance is. This sounds like a story of curiosity, persistence, and the ever-evolving nature of science. The mystery of insulin is what matters today on Discovery Matters, brought to you by the good people at Cytiva.
My name is Jeppe Sturis. I work as a pharmacologist in Novo Nordisk in Denmark, and I worked there for 29 years. Most of those years I've been a pharmacologist.
Novo Nordisk is well known around the world for being a leader in helping diabetics live with diabetes healthfully. Insulin has been a complete life changer. Jeppe's team have hypothesized that there's another mechanism underlying insulin resistance, which has not yet been considered.
I was invited to a meeting some of my coworkers had organized where they had discovered this chain splitting of other insulin molecules. And I got the thought fairly quickly that, wait a minute, if this happens to endogenous insulin, this might actually be a mechanism of insulin resistance. Maybe it happens a lot. Maybe it happens to varying degrees. With me working with insulin on a daily basis in my company, I have a great interest in the molecule and think it's still a fascinating molecule that we've known now for somewhat more than 100 years and still continues to surprise us.
Why is it such a fascinating molecule?
It has a special structure. It is not just one big molecule. It is made up of two amino acid chains that are connected to each other by two disulfide bonds.
What we found is that these bonds can shuffle around their positions so that they no longer connect the two chains. And if that happens, the insulin molecule is basically destroyed. And that's what we call chain splitting. We see in vitro that there's more chain splitting of insulin when the environment has a low redox potential than when it has a high redox potential. We estimated that a surprisingly large proportion of the human insulin that we infused to these rats was broken down by chain splitting. These data suggest that what is happening physiologically is that a substantial amount of insulin that is secreted disappears before it even has the chance to activate the insulin receptor on, for example, muscle and fat tissue. We hypothesized that a mechanism of insulin resistance might be that in some people more of the secreted insulin is broken down on its way to those insulin receptors than in other people. That would mean that the pancreas of those people with more chain splitting, they would need to secrete even more insulin to compensate for this extra insulin lost. And that would really result in elevated plasma insulin, and that's what we call hyperinsulinemia.
Hyperinsulinemia.
Can we both practice?
Yes.
Hyperinsulinemia. Hyperinsulinemia.
Good.
I knew what that meant straight away.
You did better. And it is a condition in which there's too much insulin in the blood. It's often linked to insulin resistance and type 2 diabetes.
I'm curious about how this hypothesis has challenged industry assumptions about chain splitting or about insulin resistance. How did Jeppe and his team present this new theory?
We're not saying that cellular insulin resistance doesn't exist, because it clearly does, but we're saying that there might be another player here that could be equally important. The fact that we actually estimated that it's a fairly large proportion of insulin that's lost, that's actually new. That wasn't known until we discovered it. And so if that is actually variable between people, that's when we're saying, okay, this actually might work in the same way as cellular insulin resistance would. So how do we know how much of insulin resistance is due to cellular insulin resistance and how much is due to variations and how much insulin is lost? That's basically what we're saying. This we need to find out, because nobody has looked at this for all the 100 years that we've looked at insulin, and maybe this is an important player.
Our industry is, by nature, innovative, but it is also, and rightly so, conservative, because patient safety is at the heart of everything we do. What has the conversation then been within the scientific community about this?
It's been met with surprise and also sort of a healthy degree of skepticism, because it's hard when you say there's something other than what you have been working on for many, many decades, maybe. That's a little bit of a hard thing to swallow. Sometimes people have come and say, well, it can't really be what you're saying because we've already shown that it's something else. But what we're saying is that our hypothesis doesn't exclude other mechanisms. And in the same way, we don't think other mechanisms exclude our hypothesis. I think that's important to think about. We're not saying that it's not important what's happening in cells. Of course it is. But I think also most people would agree that we don't have the full understanding of insulin resistance. And we think that this chain splitting could be a part of the full picture. So we're kind of hoping that people, especially experts in the field of insulin physiology and glucose metabolism, that they will show an interest in it and think maybe there's something about this, and that they'll want to investigate.
This hypothesis could change our entire understanding of what regulates insulin.
Instead of only looking at cellular responses, maybe we should look at understanding how insulin is degraded before it even gets to the receptor. There's actually quite a bit of literature out there that is sort of confusing and people didn't understand, and that we can actually now sort of explain maybe with this hypothesis. So one of the important things is that we see that this chain splitting is regulated strongly by redox potential. And so when redox potential is high, there's little chain splitting, and when it's low, there's a lot of chain splitting. They haven't really connected the dots, but I think we can now say, okay, an explanation might be that when you have high redox potential in plasma, there's not as much insulin degraded before it can act. On the other hand, if there's low redox potential, there's more that is broken down.
So if I'm getting this right, the redox potential in a plasma is essentially determining how much insulin gets broken down before it reaches its receptor.
Yes. When redox potential is high, there's less chain splitting, meaning more intact insulin molecules are available to bind to receptors. But when it's low, more insulin gets degraded before it can act.
That's really interesting. It's almost suggesting that insulin availability isn't just about secretion, but about how long it survives in circulation.
Right. And this process is dynamic. Redox potential fluctuates based on various factors like metabolic state, stress, or even inflammation.
So theoretically, if we could modulate redox potential, we might be able to influence insulin stability and availability.
That is a possibility.
In our case, this means that the reducing and oxidizing molecules in the environment of insulin, is like the blood, they can interact with these bonds, the disulfide bonds of the insulin molecule, and cause them to shift their positions and lead to chain splitting. And once that happens, the molecule is gone. This environment of redox is clearly important to what's happening here. And so to understand how that regulates chain splitting, I think, is what we should be looking at.
This has been the culmination of 3 years of research. So how did the team react when their paper was finally published?
There was also a reaction from the greater community.
Well, it felt good to get it out because actually we've thought about this for 3 years now. And the reaction was mixed. Some were surprised and some were enthusiastic and some were skeptical. I've had comments from former colleagues that have mainly been, wow, this might be true. Then the next step is to try and figure out what's happening. And that will take experiments. And these experiments might not be so easy to conduct. Our experiment was difficult in some ways because we had to employ special techniques to even measure these degradation products. The problem is that these degradation products of insulin, these split chains, they disappear from the body extremely quickly. And so that's probably why nobody has discovered them before, because they basically are gone within a minute. And we require special techniques to detect them.
Now, how could Jeppe's work change the way that we understand diabetes and the way that we understand insulin in its treatment?
It might be too soon to answer that. This is still a hypothesis. If it is true that increased degradation of insulin plays a role in insulin resistance, then one possibility would be to try to find out how to reduce that degradation.
Doing more research into how redox potential varies and how it's regulated within the body is important. One of the things we don't know is, we can see this happens in the body. We don't know where it predominantly happens. Does it happen mainly in the liver? Probably happens a lot in the liver. But does it happen near the muscle tissue? Does it happen near the fat tissue? This is a key question to answer. So there's a lot to still unpack with or discover with this. If you think about it, we have now 2 publications on this suggesting that this may play a role for insulin action, this chain splitting. Typically, when there's a new biological mechanism, there'll be hundreds or thousands of papers that sort of all provide new information that builds to the picture. So it's still pretty early, I'd say, but I think we're excited about what we discovered here.
As these molecules disappear almost instantly, special experiments would need to be devised.
One of the things we did was we infused a high rate of human insulin to say, okay, if it happens, the higher infusion rate we use, the greater chance we'll have to be above the detection limit of these free chains. And then to quantify the amount that is degraded in this way, we also have to know about the kinetics of the free chains. And to do that, we synthesized these free chains in the lab, injected the bolus into the rats, and saw how quickly it disappears. And then when you have both the levels from the experiment where insulin is infused and the kinetics, then you can basically estimate how much of the insulin that's infused is actually graded into these free chains. It doesn't tell us where in the body it happens. If it happens near the muscle and fat tissue, maybe we don't even see that because those free chains may never return to the plasma before they are degraded. We just don't have the measurements of the chain splitting in those experiments yet. And so some of the publications out there might benefit from being repeated with measurements of free chains.
We've talked about understanding insulin resistance, but what's the consequence of it?
Insulin resistance is a risk factor for type 2 diabetes. And the problem is when you have insulin resistance, your beta cells have to work overtime. They have to produce more insulin. When the beta cells can't do that any longer, when the insulin resistance has been there so long that the beta cells are weak and they fail to produce enough insulin. This is where this is also exciting because if indeed there is this high degradation, if we can reduce this degradation, we'll also reduce the burden on the beta cell to produce more insulin. And in some ways, we could maybe prevent type 2 diabetes if we can figure out how to reduce this chain splitting. Of course, that requires that we can show that chain splitting is a relevant element in resistance. But the exciting thing is that there may be a whole new avenue to actually understand and combat insulin resistance. After having worked on insulin resistance for basically 100 years, we still really don't have a good way to treat insulin resistance. I think most people would agree with that. There's a room for better treatments, and that may be a long time away, but 100 years, hopefully not that long. But I hope we can get wiser sooner than that, because I'd like to see some developments in this field.
Back in 2021, we discussed insulin for the first time on our podcast with Diamyd Medical. The Swedish clinical stage diabetes company discussed their precision medicine therapy for type 1 diabetes. Now we have an update on how they're advancing the precision immunotherapy for type 1 diabetes toward an FDA-supported phase 3 study.
Diamyd Medical's objective is to bring to market a precision medicine to help preserve the insulin production in type 1 diabetes patients.
And in this case, right now, particularly focusing on patients between a certain age, so younger patient population, that's the start. And then the ambition is also to expand that to a larger patient population. So right now it is type 1 diabetes, stage 3, as we call it, but then also to potentially be able to expand it to earlier stages where you have more sort of your insulin production left, so stage 1 and stage 2. And then also really important is to be able to expand it into what we call a late onset.
Meet Karin Rosén on the board of directors at Diamyd Medical.
She's articulating a huge ambition there. Jeppe was just telling us about type 2 diabetes. Big difference here is that Diamyd Medical is looking at the different stages of type 1 diabetes and how to prevent it.
Research is, first of all, the understanding of what actually triggers this disease, but also more importantly, how can you preserve this instead of just replacing what's missing? And that's where where research is really important right now. It is to develop therapies that can help preserve the insulin production, the body's own insulin production. And there we have seen major advancement. And this is particularly important in type 1 diabetes, because that's also where we see a lot of younger people getting diabetes that then causes a lot of body damage.
When we met Diamyd Medical's CEO, Ulf Hernelius, they were at phase 2B. What's happened since 2021?
Well, in short, they're in phase 3 and looking optimistically with regulators and a step beyond that.
The FDA has now given us a green light, or given Diamyd a green light, to get what we call an accelerated approval. And this is really important because then the FDA is considering this treatment so important that they are helping this drug to gain earlier access to the market by giving what we call an accelerated approval. And this gives you the chance to have an earlier read on what we call a surrogate biomarker, so not the clinical endpoint, but a surrogate marker. And this is really important, and this is where Diamyd is leading the path in the world.
At this point, in all the talk about diabetes, I started to think about a topic that is in a lot of the headlines lately.
Are we going to come up on GLP-1s by any chance? Our weight loss drugs?
That's right. And I thought, aren't those helpful for diabetes treatments and perhaps prevention? And Karin had a great answer here.
So I think that, first of all, the world of GLP-1s have taught us one thing, which is that obviously they are approved for type 2 diabetes and not necessarily type 1 diabetes.
Our focus is on the autoimmune, on the type 1 diabetes. It's not like on the weight loss or anything, but we still want to preserve beta cell function. Since if we can do that, we can have a better blood sugar control. And as Karin said, then we won't have all these bad complications that most of the time happens later on in life when you have type 1 diabetes. So it's very important.
Hold up, new voice. Who's this? Who's jumped into our podcast?
That is Sophia Mayans. She's the director of Diamyd Medical's business development, and she is helping open the manufacturing facility in Umeå. And in full disclosure, Cytiva is supporting Diamyd with our Flex Factory solution there so that Diamyd can self-manufacture the therapy they're developing. Since we had Sophia on the line, I asked her about another industry trend, which is manufacturing close to the point of the customer. And sometimes we call this in-region, for-region manufacturing.
It's amazing for us living here up in the north that we can actually work in the drug development business. It's not that many companies around where we can do that. It's also very rewarding for everybody working at the facility to know that we are actually manufacturing the drug substance that will be the drug product that will help numerous people that are at risk of getting type 1 diabetes. So that's amazing. And I mean, my role at the facility is actually to do some business development. And that might sound very weird since, I mean, this facility is owned by Diamyd Medical. The focus is to produce our drug substance. But as the facility is set up, we will have some overcapacity. So that's why I'm there to work, because I will look for collaborators that want us to manufacture biologics.
What we want to get to is prevention. That is what we want to see in 5 years. You prevent disease, not treat when they have already occurred.
And you know what they have a need for?
Do they have the need for speed?
Indeed, they do. And for that, they focus on collaborations with large pharma companies, other industry partners. Here's Karin again.
With resources, combined resources, you can actually have access to marketing muscles, distribution networks, which is also important to help get this drug faster to the patients. At the end of the day, it is the patients that needs this. And we need to make sure it's our responsibility to try to make sure that if we have an approved product, that patients, not only in Europe, but US and globally, get access to as fast as possible.
So what's ahead for them now?
I really believe that Diamyd Medical has a very exciting year ahead. So we are the only company in the world with a precision medicine for type 1 diabetes in clinical phase 3, and that is really amazing. And we also had this early readout that Karin was talking about in about a year. And for this readout, we will use C-peptide as a primary endpoint. And this is very exciting since this is a biomarker that hasn't been used for as a primary endpoint before. But it will really show us how we can preserve the beta cell function, since C-peptide is a measure on how much insulin the beta cells are producing.
Today, we uncovered a bold new hypothesis that could reshape how we understand diabetes and insulin resistance, suggesting that insulin loss might be happening before it even reaches its target.
It is a powerful reminder that science is always evolving. Jeppe and his team are pushing the boundaries of what we thought we knew about insulin, and while there's still so much to explore, their findings could open up a whole new avenue for diabetes research, treatment, and the patients.
The journey to discovery takes curiosity, persistence, and a willingness to change long-standing assumptions. If Jeppe's hypothesis holds true, it could maybe even help prevent type 2 diabetes in the future. After speaking with Diamyd Medical, we understand how type 1 diabetes progresses through 3 stages before full disease onset, with early intervention offering the best chance for prevention. Traditionally, the treatment has focused on insulin replacement, but this new precision medicine approach aims to preserve beta cell function. The future of type 1 diabetes management lies in prevention, not just treatment.
And now it's time for our favourite segment of the show, Every Day is a School Day. Okay, so every day is a school day. Dodie, what have you learned this week?
Well, this is about AI, and this is, I think, going to knock your socks off, because I know we all know that AI is about crunching big data and making it easy, but check this out. A team of U.S. scientists, including those from the Arc Institute and NVIDIA, the big AI company, they developed a publicly accessible AI model trained on the DNA of over 100,000 species across all 5 biological kingdoms, and they were looking for insights into disease-causing mutations and genome design. So this model was trained on more than 9.3 trillion nucleotides.
That is a very big number of nucleotides.
Huge, and it can predict pathogenic mutations with 90% accuracy for genes like BRCA1. Researchers believe that this model will advance drug discovery and gene therapy development for targeted treatments, hopefully with fewer side effects, and it is just a huge advancement in generative biology and using AI.
And I just wonder whether or not we're going to be able to ingest this huge new number of potential drug candidates, therapy candidates, as AI generates all of these highly accurate potential possibilities.
It's incredible.
Extraordinary. Well, I've looked at something slightly different this week, outside the area of health, but maybe it's about world health, and this is about stem cell-driven conservation, specifically rescuing the giant panda from, what shall we say, its reluctance to procreate effectively?
Oh my God, I used to love giant pandas when I was a kid. This was when Ling Ling and Sing Sing were at the Washington, D.C. Zoo.
Yeah, but Ling Ling and Sing Sing didn't do their thing.
I know, they did.
And that was the problem.
So how is this study going to help The next generation
The
of
next generation.
Ling Ling and Sing
So
Sing.
Ling Ling and Sing Sing's things are going to be supported, let's say. So look, giant pandas still face persistent habitat threats, and genetic diversity in the species is really low. And a study in Science Advances has really detailed the reprogramming of their skin cells into induced pluripotent stem cells, which can then be differentiated into gametes, the reproductive cells.
So from
Now,
the hides of the pandas.
yes, or
they
from
can-
not the hides of the pandas, but from the skin of living pandas.
Whoa.
So we don't need to skin Ling Ling and Sing
Okay,
Sing.
yeah, let's not do that.
The breakthrough, led by the researchers at the Smithsonian, it just bypasses the species' poor reproductive rates and enables in vitro generation of sperm and egg cells. And then what that means is you can have long-term viability of in vitro fertilization of the species, and you don't need to actually use gametes taken from, male and female pandas. You can actually create the gametes in vitro.
That's amazing.
So the platform could help bolster genetic diversity, but also facilitate disease modeling to address panda-specific pathogens. Try saying that fast. And things like canine distemper virus, which apparently they have. But they're also looking at adapting it to other endangered species like the Sumatran rhino and the vaquita porpoise. So there you go. Stem cells and conservation.
Amazing. Get in touch with us at discovery.matters@cytiva.com. Our producer is Beth Armett-Brewster. Editing, mixing, and supervision by Tom Henley. My name is Dodie Axelson.
And I'm Conor McKechnie. Make sure you rate us on Spotify or whichever platform you use. We'll see you when we come back with another episode of Discovery Matters.